All right, thank you, everyone. Yeah, as Jeff said, I'd like to discuss the logistics and practical difficulties of running multi-site and national research projects. Can everyone see the slide now just fine? I assume you can if there's silence. So we have Dr. Lynn. Dr. Lynn is the Interim Chief of Department and Emergency Medicine at St. Michael's Hospital, Unity Health, Toronto. He's an associate professor and clinical scientist in the Division of Emergency Medicine and Department of Medicine at the University of Toronto, and he's an adjunct professor at the Department of Physics at the Toronto Metropolitan University. Our second discussion today is Scott Haig. He's a research logistics manager for BC Resurrect, and he's been a primary care paramedic with BC Emergency Health Services for 9 years. He's heavily involved in provincial and national research initiatives, in addition to special projects within the ambulance service. And the last person discussing him today will be Alix Carter. She is a research director at London Health Science Centre / Western University Emergency Medicine and the Southwestern Ontario Regional Base Hospital Program. She's the lead investigator for paramedics providing palliative care programs. So as we said today, we've got a short presentation, 30 minutes. To start off will be Dr. Steve Lynn discussing high-level epidose discussion. After that, Scott Haig will be discussing BC research implications and logistics. And lastly, Dr. Alix Carter will be talking about palliative care research implication, experiences, and logistics. As with all the other discussions, there'll be questions at the end. So now I'm going to hand it over to Dr. Steve Lynn.
Great, thanks so much, Shane. So it's my pleasure to be able to introduce the trial called epinephrine dose optimal versus standard evaluation, or the EPIDOSE trial. This has been a long trial in the making. And so maybe I want to just go over how we came about to this trial today and kind of the process that's gone through. So as clinicians, we're really well positioned to come up with the first research questions. And so this is really what we were dealing with a number of years ago. Sorry, Shane, can you go back to the other picture?
Yeah.
Was that we looked at what we were doing for cardiac arrest, and I think we all felt that we were just giving too much epinephrine to patients. We didn't have a ceiling. We were still giving 1 milligram every 3 to 5 minutes. And overall, we thought that was a bit too much. And as we were discussing this, next slide, please, Shane. This is the Paramedic 2 that trial came out. And so this is a UK trial that had over 8,000 patients randomized to getting epinephrine by standard protocol of 1 milligram every 3 to 5 minutes, compared to a full placebo arm, which was not getting any epinephrine whatsoever. And what they found was actually quite surprising to us, that they found that there was a survival at 30 days. There's a higher survival rate at 30 days compared to with those that received epinephrine compared to placebo. But they didn't see any differences in the secondary outcome of favorable neurological outcomes at 30 days. Next slide, Shane. What was most interesting here was that for survivors who received epinephrine, 31% of them had moderately severe or severe disability. Can you just hit enter, Shane? Compared to only 18% that received no epinephrine. And so it came up to the mind where, are we actually causing some harm as we're trying to restart the heart? Are we causing some harm in these survivors by giving them epinephrine to begin with? And this really helped solidify the idea of brain resuscitation. And so instead of just focusing on restarting the heart, next slide, we thought that we needed to focus on resuscitating the brain and ensuring that we're not actually damaging the brain at the same time as we're trying to restart the heart. Next slide. And one way we approached this is we actually went back to our own animal. So what we did was taking a look at our own pigs and looking at some pig animal work. We found that brain oxygen levels actually increased with increasing doses of epinephrine, and increased the most significant after 2 doses. But what's interesting was that we were able to find that brain cell metabolism decreased after the 3rd dose, despite still maintaining a high oxygen level around the brain. Next slide. And this kind of coincided with our clinical data, which we found that the probability of survival decreased with increasing doses of epinephrine. Can you just hit enter, Shane? So from 0, 1 to 2 to 3 milligrams, and when you hit the 3 milligram mark, the probability of survival really did bottom out, and there's no differences. And in the pre-hospital setting, the average dose that was being delivered to patients was 3.5 milligrams. Next slide. And so with the data that we had, we put together a grant application. We were successful in getting CIHR funding, and we were well supported by the infrastructure that was ready with CanROC, which were the main Canadian sites that were part of the North American log. And so the Epidose trial was actually born. Next slide. So the study design is actually rather simple. So if you had a VF or VT in a cardiac arrest, you were being randomized to receiving either a low dose, which we call an optimal dose, so up to 2 milligrams, compared to the standard dose, which we defined as up to 6 milligrams. Next slide. And so if you had a VF or VT on paramedic analysis or had an AED shock that was witnessed or administered by paramedics, you either received kind of low dose epinephrine versus standard dose epinephrine. Next slide. The outcomes were very similar to what was previously published. Our primary outcome is survival hospital discharge, which is easier to get. Our secondary outcomes are looking at survivors and trying to see whether they survived up to 1 year. And what's more interesting is that we're including neurological function at 1 year, as well as global quality of life at 1 year, which it's been more recognized that there are many more issues that people have than just their daily activities. That's that the quality of the life that they had, and we're trying to capture that in this trial. Next slide. So right prior to launch, COVID hit the world, right? And so we're all affected by COVID, and priorities changed. So as the risk to performing CPR significantly increased, research around this area was put in the back burner, and the priorities of keeping paramedics safe was rightfully put as the main priority here. So even despite the pause of the actual trial itself, many things affected the trial, including kind of drug manufacturing. So during this time, drug manufacturing changed, and we lost the ability to have placebo syringes. So we actually removed placebo syringes out of our protocol and out of our study kits along the way, even before launching the actual trial itself during COVID. But as time moved on and things improved, we were able to get paramedics on board again. Research came on board, and we've since launched the trial. But what we found was that what was important is that we need to provide paramedic agencies different timelines on when they could actually get restarted. Some required more logistic changes, as well as kind of the administrative changes that need to occur prior to launch. So we need to make sure that we gave that ability for different agencies to launch at different times. Next slide. We also found that it was important to give a kind of a run-in phase during the actual trial itself. So despite going through this, having much experience in running large research trials, it became apparent that different agencies required different things along the way and came up with different issues. So having a 3-month or 6-month run-in time, you get to learn about all of these little issues as well as addressing them, either with education or even protocol changes. And even to this day, which we've launched, this is our 1 year mark since our launch. We're still learning things along the way and still making minor protocol changes, which Scott will be able to discuss a little bit more. Overall, the Epidose trial, this is really the first RCT that's looking at low-dose epinephrine compared to standard of care. This is the largest cardiac arrest trial that's going to be run in Canada. And of course, we talked about this before, that we need to really allow time for paramedic agencies to launch. And again, we're still having paramedic agencies coming on as we go on, as we get more, as time moves on. So, which has been wonderful. And with that, I'll pass it on to Scott.
Perfect. Thanks, Steve. And thank you, Shane. First and foremost, great to be here. Thanks for having us. Jeanette, thanks so much for all of your work in the background and to the executive. Scott Haig, I'm coming to you from Vancouver, the unceded and ancestral territories of the Musqueam, Squamish, and Tsleil-Waututh Nations. So thanks for having me. Steve, fantastic introduction. Thank you. This is sort of setting the stage for pretty much what I'm talking about is logistics. And I recognize a few names here from a couple other committees, so I'm glad you're here. Bit of a high level. So my role in this has been really, I call myself a paramedic research liaison. It's kind of a self-coined term, but it's come out of this long history in BC Ambulance. I'm really going to focus on BC here today, standing on the shoulders of people like Rob Schlamp, Jenny Helmer, our good friend Ron Straight, late Ron Straight, and some of these big trials that have happened in the past. So we are not new to research in BC, but as everybody knows, BC Ambulance has only been around for 50 years, coming up next month, and so we're still in our infancy, learning how to do research projects, learning how to best support paramedics. But some of the research that I've been a part of is the pre-hospital ECMO enrollment, eCPR, CORSIP, as Steve kind of alluded to, which was a COVID serology antibody study done throughout COVID on paramedics, and then now Epidose. But my role in this slide really is my least favorite slide of them all, but my role is really pretty much to manage those studies as we bring them to the front line, to paramedics. And I think it's a unique role. I think it needs to be expanded on in different sites. I think I'd really like to hear other people's questions and how their experiences have gone, taking on a similar role, as I see people here that do that. So please use this sort of time as a question gathering period and ask us questions at the end. Perfect. Next slide there, Shane. Thanks. So really in BC, as I mentioned, we focused on some pragmatic research. And I love that term, pragmatic, because, paramedics are practical beings, in my opinion. I've been a paramedic for just shy of a decade. And research that, affects paramedics, impacts paramedics, but also contributes to the progression of our service is really what I've tried to surround my research focus in. And what I've found a passion in is the logistical implementation. And really what that means is taking our history, understanding the services and the people and how they go about their business, and finding a way to marry research, high level methodologies into the paramedic frontline day-to-day practice. Without paramedics, we don't see patients. Without patients, we can't gather the data we need. And so what I've really tried to do is take this paramedic liaison role and harness my understanding of our leadership structure, and then communicate that to which we're trying to study from our research principal investigators, and then bring it to the paramedics and actually gather that data effectively and efficiently. As Steve has kind of alluded to, there's not a one-size-fits-all to every study. And sometimes we do need to change those protocols, just modify them slightly. And I'll give a perfect example that relates specifically to Epidose. We're in the process of transitioning from the way we started, which was we had epinephrine in a box, almost like a Kleenex box. We had that going onto ambulances. Paramedics would open that at the patient's side during a cardiac arrest, and they would see there's either 2 doses of epinephrine and they give that to a max, or they give a maximum of 6. We've heard over the last 6 months feedback from the front line, and we have actually taken that feedback, being like, well, this box is no longer needed. We don't have this double-blinded placebo-based trial. Perhaps we could just use an envelope and use each site's or each organization's standard epinephrine preloaded vials. So we took that feedback, and for the purpose of making it easier for the paramedics, well, honoring our research question, we've made an amendment to our protocol and to the Research Ethics Board, and we're moving into envelope-based randomization and enrollment at patient side. And so really what I see this is I see this as a perfect example of, being flexible, but also it's a perfect example of how paramedic research may differ slightly from that of, very standard formal academic research. There's nuances with our group. And so I will dig into a couple other little examples about how BC has specifically might be slightly different than the average organization across Canada. But I really want to focus on just, putting that seed, planting that seed in your guys' mind to ask questions about how things might be different, some of the hurdles that we've seen and overcome for our question period. The last little bit I want to just chat about is, that site-specific nature of BC Ambulance and Epidose here. We have a very interesting model in Victoria and over in Burnaby and Vancouver, where we have paramedics doing a ready-to-respond model. We have a model where paramedics will just get into the first ambulance that is available, and it will already have been, I guess, made ready. And so this was a bit of a challenge to ensure that we had our epinephrine in multiple cars, not just the single standard car. And now we're moving to the envelope based, not just one car with one envelope. Yeah. So outside of that, I don't want to take up too much more time. I know we've got Alix, who's got a lot more to talk about on sort of more of a national level. This is just my experience. I would very much like to encourage everybody to ask questions. Have a look at what BC Resurrect is here in the link. And if you have any questions, feel free to email me. I think we could talk about this for a whole presentation. And so I look forward to chatting more about it. Thanks, Shane.
All right, next we're going to move to Alix Carter here. Go ahead.
Okay, thanks. So as mentioned, I'm going to take this more from an overarching kind of primarily coming from the perspective of launching the Paramedics Preventing Palliative Care across Canada. I can sprinkle in a little bit of things we've learned from other trials, but more kind of a lessons learned approach. The next slide. So as I mentioned, primarily coming from this paramedics and palliative care perspective. So we launched this program in Nova Scotia and Prince Edward Island, kind of started developing in 2011 and went live in 2014. And then we're fortunate to be supported by the Partnership Against Cancer and Healthcare Excellence Canada to spread this across BC, Saskatchewan, Manitoba, Ontario, New Brunswick, and Newfoundland between 2017 and 2022. So that's the main kind of examples I'll be giving is our experience in doing that. So next slide. So the biggest overarching thing that we learned was the importance of learning to speak the same language. And that involved both educating about the EMS world, the paramedicine world, the emergency services world, but also being open to learning about the palliative care world and realizing, in fact, that was one of my first epiphany moments when we got a letter from a physician asking for someone to be placed on our paramedic palliative care registry, was that they wanted things that just weren't possible. They were asking us for lorazepam for their patients. Well, we don't have refrigeration. And so learning about each other's worlds was incredibly important. The next slide. Some of the other biggest challenges, and this was especially a big issue for us around CanRock, was data definitions. And so we had a lot of work to do in Nova Scotia around data definitions. That was one of our biggest challenges, was a room full of probably 25 people working on this, and then months and months of work afterwards. When you say, what's your call time? Did you mean E911 or did you mean dispatch? And time to care is an important measure of survival, right? And so all those definitions had to be aligned, and that was huge. And the bigger one, even than that, probably was that, as I mentioned, the institutions just don't have a lot of experience with paramedicine, and they ask for 30-page consent forms, and they ask for consent even though most of the population you're studying has died. Well, that's very impracticable, and the number of times we had to explain that was incredible. And even pragmatic research, it's just not the way a lot of institutions are used to thinking about research. So you have a lot of time explaining and learning their language. The next tremendous one is drop the dime. It's not a dime anymore to make a phone call, but make a phone call or go see people, go meet with people, have a Zoom meeting, get on a plane if you have to. And so the example I'll give for that, we go to the next slide. When we launched Nova Scotia and Prince Edward Island in 2014, we had quite a bit of discussion among all the nursing unions across Nova Scotia and PEI, and a lot of concerns about what this would mean. Duplication of service and occupational territory and professional identity. The nursing union on PEI was actually prepared to launch a grievance. That's when I got on a plane and I went to PEI because this was important enough that we needed a face-to-face meeting to sit down and understand, speak each other's language, understand what the fears were and the concerns were. This table that you see here is just the in-project knowledge translation, knowledge exchange that we did just in Nova Scotia. This isn't the end of project conferences. This is just the go meet with people. It's a lot. You need to go meet with people. And then if you can advance it one more, one of the other things that we learned, it was about understanding what the right message would be for the particular people we were talking to. So was it about professional identity, or were we talking to a minister who really wanted to know about cost, or an operator who might want to know about time on task? And understanding which piece of that to emphasize when we were meeting with different people, and also who the right messenger was. So sometimes it was me, and I would go to the meetings as the medical director, as the research principal investigator, as the physician, that people would want to hear that perspective. But sometimes my co-lead, who's a social worker with psychosocial oncology, would be the one to go to the meeting because she was the right person to be the messenger. And sometimes it was less threatening for our research coordinator to actually be the one to be in the meeting and really be listening and understanding perspectives. And sometimes it was the right moment, like a lot of our media interviews were actually done by some of our paramedic team members. And a lot of our media presence was paramedicine and family members. So the right messenger is also really important. The Shawshank Protocol. So I full credit to Andrew Travers for this one, but I don't know if you remember when the main character here gets assigned to the office. He had pulled the admin duty, and he really wanted a record player, I think, and so he sent a letter. And he sent a letter every week until he got it. And so that was always one of the things that Andrew told me was you just keep asking. You don't take no for an answer. And I don't know if that makes me foolish or stubborn or just persistent enough. Sometimes you just have to keep asking, and you keep understanding their message, and you keep listening, and you keep learning and going out and meeting with them. So the next slide here. So in our first project, this is how we learned this one the hard way. And I would say, I wish I could have done this one differently. We were funded by CIHR to do a comparison between Nova Scotia and British Columbia. And so British Columbia was our control at the time. Nova Scotia had implemented and BC had not. And we were trying to prove that it was in fact the implementation of the program that had made a difference to how much time people were spending at home and the patient and family experience of that end of life care journey. And we were told that you couldn't share data between provinces. And I have since learned that is not true. And I wish that I had tried the Shawshank Protocol at the time and just kept sending letters and not taken no for an answer. I certainly since then have not taken no for an answer, because I think it would have been a much better project to really be able to prove mathematically and do the right analysis. So don't take no for an answer. Keep asking. You can advance it. Some of the other things we learned, this was CanRock. So if you look at this, we first started our CanRock application in 2016. And I think that's really just when we put it on paper. I think we were talking about it even before that. And our privacy people said, you can't send data out of province. You can't access that data for research. That's quality data. You can't link to hospital outcomes. And we met over and over again. And if you click, I think once more, there you go. You can see that our signed data sharing agreement was signed in the fall of 2022. So that's 6 years. Yeah, so you can send data inside the country. There are situations where you could send it outside of the country, but certainly there are a lot of regulations around where it's going and under what circumstances and what data is divulged, definitely. But you can share data with other provinces and with other agencies, and CanRock is proof of that. So it took, again, speaking the same language. One of the other things that I think has been tremendously helpful is to make lots of friends. I mean, first of all, it's just lovely to have a community like this where you can come together and talk to each other and learn from each other. But there's some practical examples where it has really saved us to know people that we could call upon in different places. So on the next slide, there's a couple of ways that it can happen for sure. You should definitely anticipate that the person you've initially made contact with to do the study at a given site may move on, because that happens a lot. The example I've given here is British Columbia, but the same thing happened in Newfoundland, the same thing happened in Prince Edward Island, the same thing happened with the services in Ontario. So it's a really smart idea to have multiple contacts, and then to just have a few friends that you could always call and say, who should I talk to about this? And definitely, so, initially we were talking with William Dick, but then it moved on to Jenny Helmer. Initially it was Doug Andrusik, and then there was a lady named Sandra, and then Nichelle Wall, and then Nick, and then, so you get the idea. People move on, and they get different jobs, they get promoted, they have different interests. So it's good to have multiple contacts in different places. And then that's reciprocal, right? So if you get asked by one of your friends to do something, then you find a way to say yes and help them out too, right? So we help each other, and we all support each other and lift each other up for sure. All right. Some of the other, the subset of that was definitely try to make friends in high places, because we had to get regulation changed on Prince Edward Island for the implementation to work. And so being able to work your way up to having the deputy minister be supportive of your project, and then to talk to the other deputy ministers in the other provinces and tell them that this is a really good idea and they should really do it. We recently, here in London, where I've just recently relocated, almost couldn't participate in something until the VP Medicine said, this would look really bad if we didn't do this. So don't be afraid to reach out to those people as well. And then one of the other kind of big things that we learned was that time flies. And so the more you can find time efficiencies, the better off things will be. So I have a few ways that we learned that, a few examples to illustrate ways that you can do that on the next slide. One was finding out that there now, thankfully in Ontario, is the development of this Clinical Trials Ontario group. And similarly, slightly less recently, moved to a provincial research ethics board in Nova Scotia. So find out if there's some kind of umbrella organization you can get approval from, that you don't have to go to each one over and over and over again. And failing that, find out which one is the most demanding. Do that one first. So for example, the PEI Research Ethics Board was traditionally fairly happy to kind of take the approval from Nova Scotia and review it, as opposed to requiring a complete from scratch application. So there's ways like that. Definitely feel free to plagiarize protocols. I think most people would agree to that. Here's my protocol, please copy it. But do adapt, of course, to the local requirements and differences, because there will be a few. And then one thing I wish I had learned sooner was to start my application to research ethics and to data access, especially the day that we submit the grant. Because if you wait till you get the grant, you've only got 12 months and the clock is already ticking, and it could take 12 months to get data access approval. So those are just a few ways to try to find efficiencies for sure. All right. You can go to the next slide. Yeah. So hopefully those are a few kind of more high level than some of the other things that were talked about today, but things that we learned through the implementation of our mostly paramedics and palliative care, but certainly CanRock, sepsis, Epidose, all of those trials too.
Great, thank you so much. So for more information and other questions, I put everyone's email addresses there. Yeah, I'll hand it over to Jeff, who's going to coordinate questions if we have time.
We do, and there is a few questions in the question and answer. So we'll start off with the first one from Sterling Martin. So how is oxygenation, this one's for Steve, I believe. How is oxygenation levels measured? Is there a chance of reduced CO2 and vasodilation causing negative cerebral outcomes?
Yeah, so great question. So we use broadband near-infrared spectroscopy to measure oxygen levels in the microvasculature of the brain. So specifically for pigs, we actually remove their skull because their skull is so thick, and we actually have the actual sensors right on the dura of the brain to measure as accurately as possible. And you're right, there is that possibility when you're thinking about any kind of hypotensive effects where you actually decrease blood flow or you have increased CO2 demands, changing the vasculature and the flow of the brain that can affect it. Much of the animal work, the way I see it, is always hypothesis generating. It's very difficult to try to translate what we find in the animals to humans, but at least it gives us a bit of a sense of what can potentially happen as we help kind of do any kind of study designs for any type of clinical research.
Fair enough. The next question reads as such. So from Anonymous, how much was correlation accounted for in the analysis? The circumstances under which the 3 doses were being used could affect brain impact?
So it's a great question. It's going to take a little bit more time to answer that. So I might answer that through typing, and I'm going to provide some of our research papers to help describe some of the analysis part, because I just know out of the interest of time.
Fair enough. Next question is, while there appears to be a correlation between the number of epinephrine doses and survival outcomes, were you able to establish causation? So can you establish that the epinephrine itself was the cause of less optimal outcomes, or was perhaps whatever pathology that led to the extended resuscitation and thus more epidoses that led to a worse outcomes?
Yeah, so great question again. Simple answer is no, we can't, it's hard to establish causation. A lot of the work that was previously done to help define our actual study protocol was all based on hypothesis generating work. From the kind of the animal study work, we could get at least some sort of pathophysiology and physiological measures that help provide some of the answers. But when you look at retrospective data, observational data, it's all hypothesis generating. So obviously with time, we're giving a lot more epinephrine when a cardiac arrest lasts longer. These patients are obviously sicker, which is why they need more epinephrine, and they don't have ROSC sooner. So when you look retrospectively at any of our data, again, it's very difficult to pinpoint kind of what the reasons were, why they either didn't have survival or they had poor outcomes to begin with. So really the only answer to try to answer the epinephrine question is to look at prospective data. So particularly trial data. But before we designed this, we really looked at various dosings of epinephrine, as well as modes of administration. So either boluses or infusions, see whether there are any differences that we can see from the animal work to help inform the study design.
Okay, fair enough. We have to cut it off here. If you're able to answer, so there's quite a few more questions in the Q&A. If you're able to answer, I think people would appreciate that. Thanks to everyone from the panel for that discussion.